All right, for the second one, we're going to talk about Punnett squares. And this is a visual way of trying to figure out how things are related to each other and the possibilities of having a certain type of child and what those characteristics would look like. And so what we're going to do is look at Punnett squares. For the Zoom session, we're going to work through some of these. We'll have some problems that I will want you to be able to work through, and we'll talk about. But for right now, what I want to do is kind of go over the general concept on what some of the terms mean. So this was devised in 1905, so it's been around for quite a while. And it is a way, again, to look at the possibilities of having a particular genotype, and we'll even talk about what that word means in a minute. So some helpful terms. First of all, heterozygous and homozygous. In the simplest form, it's thought for a lot of the genes is you have two copies. You have one from mom, one from dad. So heterozygous, the term means that you have inherited different alleles. So in the pea flower example, heterozygous means that you have one for purple flowers and one for white flowers. So that would be a heterozygous. So, and how that is usually represented in a Punnett square, you can see up here on the top right square would be a large B and a small b. Because those don't look the same, that would be heterozygous. Homozygous means that they match. So this little b in the bottom right, and this large B in the top left, those would be homozygous. The trait that you inherited for that character is the same from both mom and dad. And then the dominant is, again, one that is fully expressed. So like in the purple flower, purple is the dominant color. And so that means if you have one of the dominant traits, what you're going to look like is purple. And then recessive means that you have the small traits. And the only way that's going to show up is if you have both of the small traits. Okay, so going through this square, the first one top up here, if the large B stood for purple and the little b stood for white, what you would have is this organism up here is called homozygous dominant, and this would be a purple flower. The one on the bottom right would be homozygous recessive, and this would actually appear as a white flower. And then you have two here that are heterozygous, and that's usually just the term we use, heterozygous. And it's the assumption then is heterozygous means that you are purple because this recessive trait is hidden. So you would turn out purple. So again, homozygous dominant be purple, homozygous recessive, it would be white, and then the heterozygous would be purple. In this case, what you're also seeing is homozygous recessive. That means the only way to have a white flower show up is you have to have the two recessive traits, and that's typical for what we call complete dominance, where one hides the other, and we'll explain that term again in a minute. The only way to become to have the recessive trait to be that white flower would be to be homozygous recessive. All right, so another terms that I was on a earlier slide that you need to know is phenotype and genotype. These are, you got to be very careful, but these are often kind of screwed up a little bit. Phenotype is basically how an organism appears. That is the trait you're looking at for that character. So purple and white in this in the flower examples would be phenotypes. Genotypes, on the other hand, is really what does what's the information on your genes, what are your alleles saying. And so what I want you to realize on a test or a quiz or even working, when I'm asking for these terms, I'm really asking for phenotype. Describe it. What are you looking at? What does it look like? What's expressed? And then genotypes in the problems are always asking about the letters in the Punnett square. So the last one was B's. For purple and white, P would probably make more sense. So the phenotypes is large. So if I looked at this individual here and I asked you, what's its phenotype, you would tell me it's purple. But if I asked you what's its genotype, you would give me large P, large P. And again, it's homozygous and dominant. Okay, this is homozygous or heterozygous. And then again, phenotype is still purple. Heterozygous, still purple. And then for this last one, the genotype would be little p, or homozygous recessive. And then the phenotype would be white. All right, and then there's a thing called a test cross. What a test cross does is it looks at a flower and or whatever the organism is. Now we don't do this in people, but what it does is if you have an unknown, like a purple flower, technically in this situation, if you go back, could be homozygous, dominant, or heterozygous. There's no way to really know by just looking at the flower. So what you have to do is, if you're trying to come up with a true breeder, one of the ways you can do this is do what's called a test cross. And we do this in flies. We'll do this with plants. Again, we don't do this in humans. But what you do is you take a homozygous recessive, somebody who is little p, or little whatever the letter you're using, and you cross that with an unknown. And then you have predictions. If you have a dominant, in this case, purple flower, and you breed it with a homozygous recessive, if all the offspring come out purple, that means that it is probably large P, large P. It will be large P as long as you do a large enough sample. And then if you cross a heterozygous, large P, small p, with the homozygous recessive, half of them are going to come out white. And so the first white one that comes out will tell you that that is not a true breeder. So a test cross is a way of determining an unknown, whether it is a true breeder or not. All right, and then this is the other Mendel's law, and it's law of independent assortment. And this is what you're seeing here is we're looking at a couple of different traits. We're looking at the colors, green and yellow, and then we're also looking at whether they're round or wrinkled. And so here we have two traits. And what I want to point out on this one, the law of independent assortment, that's the same thing we talked about in meiosis, is how one set of chromosomes has no effect on the how the other set of chromosomes line up on that metaphase plate. So he figured this out also, not by looking at chromosomes, but looking at offspring. And so what he's showing you here in this law is that whether it is yellow or green has really no effect on whether it's round and smooth. And so that's where he came up with the law of independent assortment. Because the odds are, and we're gonna look at those numbers in the Zoom session, what those really mean. Okay, so again, it doesn't matter. Yellow and green doesn't have effect on smooth or wrinkles, so they're independent of each other. All right, and with that, we will stop this video and move on to our next one.